Hi, in this video we are going to learn the anatomy of the eyeball, the histology of the retina, and the fundoscopic view of retina with photoreceptors. So, to the nasal side you have the optic disc, and the retinal vessels, they spring out of the optic disc. So here is the optic disc, and lateral to it is the yellowish area called macula lutea. And within the macula lutea, there is a depression which is called fovea centralis. So here is the fovea centralis. Now, if you look at a person's eye through a fundoscope, this is how the structures will appear. So if we look at the optic disc and write it outside, and here's the optic disc, and within the optic disc there are no photoreceptors. So if you throw a light on it, it will create no stimulus or produce no image. So there will be no image, and it's called the blind spot, which is the physiological blind spot. Now, if we look at the fovea and draw it outside here, and this is a depression in the macula lutea, and the fovea has a higher concentration of cones. Cones are the photoreceptors. If we draw another diagram here and show the retina and the different organization of cones and rods, so in the centermost there will be cones, and they fade out to the periphery, but in the peripheral most area there will be a lot of rods. So in the center, C is for the cones and C is for the center, and then the rods, there's a lot of rods in the periphery of retina. So this is how they are organized. Now if you look into another diagram and draw in another way the different zones of retina, this is the retina, and this is the macular area or macular zone in the center, which has a lot of cones, and then next to it is the paramacular area, and the lateral most is the monocular area. If we draw a brain diagram and show the different fibers going to different areas in the occipital lobe, so the posterior most is the macular fibers going there, and then the paramacular fibers, and of course then the monocular fibers. So this is the occipital lobe, and here's the front and the eyeballs. So now let's look at the photoreceptors, the cones and the rods. Now the photoreceptors are for your vision, for the bright light and what are for the dim light, and what is for the color vision. So here we draw the cones, and a structure of cones, it looks like a cone. So here is the photoreceptor of a cone, and then area which contains the pigments, and here is the nuclear cell body of the cones. So here is the photoreceptor, and here are the pigments in the cones, and these pigments are called iodopsin. They are sensitive for the bright light, and they help you differentiate between different colors. So the cones, they are for the bright light, and then we have the rods. The rods I will draw in black. They look like a rod, and it has the same areas like the pigment area and the nuclear cell body and then the photoreceptor, and the pigments in the rods are sensitive for the lights and called the rhodopsin, and it's sensitive for the dark lights. So if there is a darkness, there will be it is like a stimulant, and it produces more rhodopsin, and rhodopsin is good for the dim light. So D in rods, D dims, right? This is what it might help you remember. So in the centermost are the cones, and then in the periphery are the rods. Now let's look at the anatomy of the eyeball. If we draw here eyeball and then show the different layers of the eyeball. So this outermost layer, it is called the sclera. And when the sclera becomes transparent on the front, it is called cornea. So this one is the sclera, and here is the cornea, and it's the transparent sclera. Okay, so it's called cornea. And then next to the sclera is the choroid layer. Choroids contain the capillaries, the blood vessels. So, and then next to it is the ciliary body, and we will show here the ciliary body, and then the iris. Here is the iris. Now, the choroid, the ciliary body, and the iris, these three together are called uvea. It's called uvea. Okay, so then from ora serrata here, the retina starts and it goes back and it makes the optic nerve. So this is your retina, and within the retina there are the vessels coming to the eyeball, the central retinal artery supplying parts or most part of the eyeball, and then the central retinal vein that takes the deoxygenated blood from the eyeball. So here is the lens and the anterior and posterior chambers. Now let's look at the retina and the layers in the retina, the histology of retina. It has different cells and layers in retina. So the first one we draw, which is the outermost layer and which is not part of the retina, is the choroid. And this choroid layer has the capillaries, which are the choriocapillaries that supplies the part of the retina. So this is not part of the choroid is not part of the retina, but it supplies the part of the retina. Okay, so here is the cells layer, and these cells are called the pigment cell layer. Okay, so these are pigment cells here, and it's also called the pigment cell layer. And now if we put the cones and the rods in this layer, so here is the rod, and again the rod has the same structure that we have just learned it. So it will have the photoreceptor region with the pigments, and we have the cones in red. Now this is the outermost area of the retina. This is not the inner side, it's the outer side. Okay, so here is your cone, and the cone has the photoreceptor region and the nuclear body. And if under the cones we have the bipolar cells, and the bipolar cells receives the information or the action potential from different cones and rods. It has connection to different cones and rods, okay, and it sends the fibers or the axons down to stimulate or give the action potential to the ganglion cells, and the ganglion cell has the fibers which together, many ganglion cells will send the fibers together, they will make the optic nerve. So these bundle of fiber will make the optic nerve. So these are the three types of cells. And now if we look at the retina and its layers, the first and the outermost layer is the pigment cell layer, and then the rods and cone layer. So this one is the layer of cones and rods. And then we have the outer limiting membrane layer. This is the outer limiting membrane layer. And then we have the outer nuclear cell layer because there are nuclear bodies there. And then we have the outer plexiform layer because there is the plexus there. And then we have the inner nuclear layer because it has the nuclear bodies in there. So it has the inner plexiform layer because there's different fibers making the neuronal synapses or plexus. Then we have the ganglion cell layer and the nerve fiber layer, and we have the innermost layer. So this is the inner limiting membrane layer. Now this is the inner side, and the light will enter here and it will go up and then the action potential will be produced. So if we look at the different cells, the cones, the rods, the bipolar, and the ganglion cells, right? Now, if we put the other cells in the retina, it will be the horizontal cell. The horizontal cell, it gives the fiber to both plexus here and there, cones and rods both. And then we have the amacrine cells. And the amacrine cells kind of also is a horizontal kind of cells. And it gives fibers to different bipolar and ganglion cells. And then we have one more cells called the Mueller cells. It extends from the outer limiting membrane to the inner limiting membrane. So this is all about the histology of retina and the anatomy of the eyeball and the fundoscopic view of retina.